previous chapter, we have seen Mendel imagined factors, which we call them genes now. Later scientists had seen chromosomes during cell division, but they didn't know with what chemical or molecule the chromosomes or genes made up of and how they look. And also, it was seen that chromosomes double during cell division. In this chapter, we will see how scientists identified chemical composition of chromosomes, the structure of chromosomes, and how chromosomes get doubled. At its core, every cell has to do some function. Who guides these cells to do a particular function? It is genetic material. Genetic material has information in it. After multiple research and theories over 100 years after Mendel's experiments, it is found that DNA is genetic material, at least in many organisms. If DNA is genetic material in majority of organisms, what is the genetic material in rest of the organisms? It is RNA. So, there are two genetic materials. One is DNA and other is RNA. If you seek full form, it is deoxyribonucleic acid and ribonucleic acid. So, DNA and RNA are nucleic acids. As name suggests, they are acids. RNA acts as a genetic material in some viruses, but RNA mostly functions as messenger, means it reads information present in DNA and give it to others. RNA has other functions as well. It functions as a adapter molecule, structural molecule, and catalytic molecule. Now, let us discuss about the structure of DNA. Two important things we see here. Its structure and how it is packed. We will start with a molecule deoxyribonucleotide. It is a monomer. Arrange multiple deoxyribonucleotides side by side. Now it becomes a polymer of deoxyribonucleotides. When you add many of deoxyribonucleotides, it becomes long. So DNA is a long polymer of deoxyribonucleotides. Now add another long polymer opposite to it. Now it is double-stranded. Deoxynucleotides lying opposite to each other are complementary. We call them base pairs also. So length of DNA polymer can be described in terms of number of nucleotides or number of base pairs. This number or length is unique for a particular organism or characteristic of an organism. For example, a bacteriophage known as Cy174 has 5386 nucleotides, that is 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 power 6 base pairs. Haploid content of human DNA is 3.3 into 10 power 9 base pairs. Let us discuss the structure of this long polymer. DNA is polymer of deoxyribonucleotides. RNA is polymer of ribonucleotides. Deoxyribonucleotides and ribonucleotides together referred as nucleotides. A nucleotide has three components. 1. A nitrogenous base. 2. A pentose sugar. Ribose is present in case of RNA and deoxyribose is present in case of DNA. Third is a phosphate group. Nitrogen base. There are two types of nitrogen bases, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, uracil, and thymine. Purines, adenine and guanine are seen in both DNA and RNA. Among pyrimidines, cytosine is seen in both DNA and RNA. Thymine is seen in only DNA and uracil is seen in only RNA. Suppose RNA is formed by reading information from DNA. In place of thymine in DNA, RNA will have uracil. Next, take a ribose. Ribose is a pentose sugar, means it has 5 carbons. A nitrogen base is attached to OH group of first carbon with N-glycosidic linkage. Nitrogen base plus pentose sugar is called nucleoside. Adenine with ribose sugar becomes adenosine. Adenine with deoxyribose sugar becomes deoxyadenosine. Same way, guanine with ribose sugar becomes guanosine and guanine with deoxyribose sugar becomes deoxyguanosine. Cytosine with ribose sugar becomes cytidine. Cytosine with deoxyribose sugar becomes deoxycytidine. Uracil with ribose sugar becomes uridine. Uracil cannot associated with deoxyribose sugar. Very important. Thymine with deoxyribose sugar becomes deoxythymidine. Thymine cannot associated with ribose sugar. Now, phosphate group attaches to pentose sugar at OH group of fifth carbon through phosphoester linkage. So, nitrogen base plus pentose sugar plus phosphate together called nucleotide. In RNA, we have adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate. In DNA, we have deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. In a polymer, two monomers, that is, two nucleotides, are linked by 3,5 phosphodiester linkage. When only two nucleotides are present, it is called dinucleotide. When three are present, it is called trinucleotide. When many are present, it is called polynucleotide. Once polymer is formed, it has two ends. At one end, free phosphate is present, that is at fifth carbon. So it is called 5' end. At other end, free OH group is present at third carbon. It is called 3' end. Sugar and phosphate form a backbone of polynucleotide chain. Nitrogen bases project from backbone. RNA nucleotide has extra OH group at 2 carbon of pentose sugar or second carbon of pentose sugar. It is not present in DNA. That's why it is called deoxy. Friedrich Mischer was first who identified there is acidic substance present in the nucleus. He named that acidic substance as nuclein. But due to technical limitation, structure of the nuclein could not be studied for long time. Then comes Maurice Wilkins and Rosalind Franklin, who subjected the nuclein to X-ray diffraction to see possible shape. On the other hand, Erwin Chargaff observed that the ratio between adenine and thymine and between guanine and cytosine are constant and ratio equals 1. That is, if adenine is in X amount, thymine is also in X amount. Same way, if guanine in X amount, cytosine is also in X amount. But still, DNA structure was not known. In 1953, James Watson and Francis Crick, based on data of X-ray diffraction and Erwin Chargaff's observation, they proposed double helix model for the structure of DNA. One of the hallmarks for their proposition is base pairing. The unique property of polynucleotide chains is base pairing. Base pairing is complementary in nature. That is, A pairs with T and T pairs with A. Same way, C pairs with G and G pairs with C. If we know the sequence of nucleotides in one strand, we can predict sequence in opposite strand. If both strands of parent DNA acts as template for synthesis of strands, then daughter DNAs will be similar to parent DNA. Now we will see salient features of double helix structure of DNA. DNA is made up of two polypeptide chains. Each chain has backbone and projections. Backbone is made up of pentose sugar and phosphate group, and projections are nitrogenous bases. Both chains have antipolarity. If one chain has 5' to 3' from right to left, other chain has 3' to 5', that same right to left. Nitrogenous bases from each strand held together by hydrogen bonds. They form base pairs. Two hydrogen bonds are seen between adenine and thymine, and three hydrogen bonds are present between guanine and cytosine. So, purine always pairs with pyrimidine. Helix means it's a coiled structure. It is coiled in right-handed fashion. One turn is a pitch. Pitch is about 3.4 nanometer. One pitch has 10 base pairs. So, distance between base pairs is about 0.34 nanometer. Plane of one base pair stacks over other and held by hydrogen bonds, which confers stability to helical structure. Explaining DNA simply as double helix structure is revolution. Soon, Francis Crick proposed central dogma. Central dogma tells about flow of information. So, information in DNA flows to RNA, then RNA to protein. In some viruses, information flows from RNA to DNA. That is called reverse transcription. Now let us see about packaging of DNA. Typical mammalian cell DNA has 6.6 into 10 power 9 base pairs. When multiplied with 0.34 nanometer, it gives 2.2 meters. See, nucleus is about 10 power minus 6 meters. How such a long DNA fits in this small nucleus? It is like fitting a sari in matchbox. Let us see how it fits. DNA exists differently in prokaryotes and eukaryotes. Prokaryotes does not have defined nucleus. Negatively charged DNA is held by positive charged proteins in a region called nucleoid. So here DNA is arranged in large loops. In the other hand, in eukaryotes, the organization is complex. The organization of DNA is complex. It has defined nucleus. In nucleus, there are proteins called histone proteins. These proteins are basic in nature and positively charged. Histone protein is rich in lysine and arginine amino acids, which are positively charged as they carry positively charged side chains. 8 histone proteins are organized to form histone octamer. Negatively charged DNA is wrapped around positively charged octamer. The structure formed is called nucleosome. Nucleosome contains 200 base pairs. Many nucleosomes form chromatin. Chromatin is thread-like structure seen in nucleus after staining. Under electron microscope, nucleosome appear as beads on string. If you see a non-dividing cell, you find a chromatin. During cell division, chromatin is further condensed to form chromatin fibers. Chromatin fibers further condensed to form chromosomes. These condensations are possible due to non-histone chromosomal proteins. In nucleus, at some regions, chromatin is loosely packed. It stains light. It is called euchromatin. At other regions, chromatin is densely packed. It stains dark. It is called heterochromatin. You just imagine where it is easy to read information. Obviously, where chromatin is loose. So, euchromatin is transcriptionally active. Now, let us see the events related to the search for genetic material. Mendel imagined factors. Mesher discovered nuclei. Other scientists narrowed the search to chromosomes as genetic material. But molecular nature of genetic material was not known. The theories and experiments related to search for chemical or molecular nature of the genetic material took place before Watson and Crick's model. In 1928, Frederick Griffith conducted an experiment with bacteria Streptococcus pneumonia and mice. He had grown Streptococcus pneumonia bacteria. Two types of bacteria were grown. One type formed smooth colonies in culture plate and other type formed rough colonies in culture plate. He called bacteria in smooth colonies as S strain and bacteria in rough colonies as R strain. When he studied bacteria in smooth colonies, they formed mucopolysaccharide layer around cell wall. Now he wants to study effects of these bacteria on mice. First, he injected S strain and R strain into two different mice. Mice with S strain died and mice with R strain was alive. S strain is virulent. Next, he killed a S strain bacteria by heat. He injected killed S strain into mice. Now, mice was alive. Next, what he did was he combined killed S strain and live R strain. and injected into mice. Two things happened. One, mice got killed. Second, he obtained live strain from this mice. Then he understood that something is passed from killed S strain to live R strain. This something was not destroyed by heat. This something changed or transformed R strain. So he called this something as transforming principle. This transforming principle could be genetic material. But still, biochemical nature of this transforming principle was not known. Initially, biochemical nature of genetic material was thought as protein. Then, Oswald Avery, Colin MacLeod, and Maclean McCarthy during 1933 to 1944 worked to determine biochemical nature of transforming principle. They took heat-killed S strain. From this, they separated proteins, RNA, and DNA. They treated live R strain with these molecules separately to see which of these can transform R strain to S strain. They found that DNA was responsible for converting R strain to S strain. They also confirmed with other way. Proteins, RNA, and DNA are treated with proteases. Now injected mixture, R is transformed to S. So this indicates it's not protein. Then all three are treated with RNAs, which destroys RNA. Then also R strain is transformed to S strain. So it's not RNA. When it was treated with DNAs, which destroys DNA, transformation not seen. Again, they concluded genetic material was DNA. This experiment and proof was not enough for many biologists to convince DNA as hereditary or genetic material. Then came Alfred Hershey and Martha Chase in 1952. They worked with viruses which infect bacteria. These viruses are called bacteriophages. How bacteriophage and bacteria behaves? Let us see. Bacteriophage or virus sits on the bacteria. Genetic material is passed from bacteriophage to bacteria. Bacteria treats bacteriophage's genetic material as its own. Subsequently, it manufactures more virus particles. Hershey Chase worked on whether genetic material passed was protein or DNA. See, DNA contains phosphorus and it doesn't contain sulfur. Protein contains sulfur and no phosphorus. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA. Similarly, viruses grown on radioactive sulfur contained radioactive protein. Now, these two types of radioactive viruses are allowed to infect bacteria. They used blender and centrifuge to remove viral coats from bacteria and separate virus particles from bacteria respectively. Bacteria infected with radioactive DNA viruses are also radioactive. Bacteria infected with radioactive protein viruses are not radioactive. This tells that DNA is genetic material which passed from bacteriophage to bacteria. Hershey and Chase experiment unequivocally proved DNA as genetic material. But subsequently, it was found that RNA is genetic material in some viruses. Example, tobacco mosaic virus and QB bacteriophage. So, there are two genetic materials, DNA and RNA. When two are there, then comparison starts. DNA is predominant genetic material, whereas RNA mainly acts as a messenger and adapter. Who explain this difference? Difference in their chemical structure explains this. For a molecule to be genetic material, it should fulfill following criteria. It should be structurally stable, chemically stable. Still, it should provide scope for slow changes, that is mutation, for evolution. It should express and it should replicate. Let us compare above each criteria with respect to DNA and RNA. Both DNA and RNA can self-replicate. Other molecules like proteins fail to do so because they cannot replicate. DNA is double-stranded. Even if each separates its strands, they come together during suitable conditions. It's even proved by Griffith experiments. There, heat did not destroy some properties of genetic material. So DNA is more structurally stable. Do you remember there is a OH group at 2 carbon position of ribose in RNA? So RNA is reactive. Even we know RNA can be catalytic. So it's reactive. So DNA more chemically stable than RNA. Thymine is seen in DNA and uracil in RNA. Thymine is more stable than uracil. Presence of thymine helps in repair. Both DNA and RNA mutate. RNA mutate at faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. Let us see about RNA world. RNA was first genetic material. There is enough evidence that essential life processes like metabolism, translation, and splicing evolved around RNA. RNA used to be both as a genetic material and catalyst. There were some biochemical reactions catalyzed by RNA. Due to its catalytic function, it was reactive. If something is reactive, it is unstable. Then chemical changes to RNA occurred, resulting in formation or evolution of DNA. DNA is more stable. DNA is stable for three reasons. One, due to its chemical nature, it is less reactive. Second, double strand make it more stable. Third, it also has repair mechanism. Now let us see about replication. Watson and Crick had proposed a scheme for replication of DNA while proposing double helical structure. Base pairing suggested there could be possible copying mechanism. The scheme suggested that the two strands of DNA would separate. They act as a template for the synthesis of new complementary strands. resulting in 4 strands after completion of replication. So now there are 2 DNA molecules. Each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. The experimental proof of semi-conservative method was given by Matthew Michelson and Franklin Stahl in 1958. Before this experiment, there were 3 competing theories on how DNA replicated. One theory tells replication is conservative. Here, original DNA stays intact and a completely new copy is made. Second theory tells replication is semi-conservative. Here, each daughter DNA molecule contains one old and one new strand. Third theory tells replication is dispersive. Here, each strand of both daughter molecules contains a mixture of old and new DNA. Now let us see Michelson-Stahl experiment. Michelson and Stahl used Escherichia coli, E. coli, and isotopes of nitrogen to distinguish between old and new DNA. Nitrogen is a key component of DNA. It is found in nitrogenous bases. Let us see step-by-step process. They grew E. coli for many generations in a medium containing N15. It is a heavy isotope of nitrogen, but it is not radioactive. Eventually, all the bacterial DNA contained only N15. They transferred these bacteria into a medium containing N14, which is the normal lighter isotope. They took samples at specific time intervals, example, every 20 minutes, which is the doubling time for E. coli. They extracted the DNA and used cesium chloride density gradient centrifugation. This process separates DNA based on its density. Heavy DNA containing N15 sinks lower, while light DNA containing N14 stays higher. The results across generations provided the definitive answer. At generation 0, all DNA was heavy, that is N15. It formed a single band at the bottom of the tube after centrifugation. At generation 1, that is after 20 minutes, if replication were conservative, there should have been 2 bands, one heavy and one light. Instead, there was only one intermediate band. This meant the DNA was hybrid, containing N15 and N14. So this ruled out the conservative model. At generation 2, that is after 40 minutes, there were 2 bands, one hybrid and one light. This ruled out the dispersive model. If it would have been dispersive model, it would have shown a progressively lighter hybrid band. So this experiment confirmed the semi-conservative model. Later, semi-conservative method was even proved in higher organisms like plant and human cells. Similar kind of experiments were conducted by Taylor and colleague in 1958. They used radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes. They performed this experiment on Vishia faba beans. The experiments proved that the DNA in chromosomes also replicate semi-conservatively. Let us understand the machinery of replication and enzymes involved. Two important enzymes we see here. One, DNA dependent DNA polymerase. Second, DNA ligase. We will see two scenarios. First scenario, small double-strand DNA fragment has to be replicated. Two strands of DNA get separated. To separate them, there should be breakage of hydrogen bonds. It requires energy. Now, enzyme DNA-dependent DNA polymerase comes and start to read DNA template. It is called DNA dependent because it reads DNA strand. It is called DNA polymerase because it synthesizes DNA strand. As there are two strands, two enzymes come to synthesize. This enzymes reads the template from 3 to 5 prime direction and synthesize new strand in 5 prime to 3 prime direction. As initial parent strands run in opposite direction, synthesis also takes place in two directions. DNA-dependent DNA polymerase catalyze the polymerization of deoxygenated nucleotides. Let us see some properties of this enzyme. It is highly efficient, means it catalyzes large number of nucleotides in short time. E. coli has 6.6 into 10 power 9 base pairs. It copies these many base pairs in 18 minutes. Means it polymerizes 2000 base pairs per second. Other property is it catalyzes polymerization with high accuracy because any mistakes results in mutation. Next property is it requires high energy. See, it polymerizes deoxyribonucleoside monophosphate. But deoxyribonucleoside triphosphates are used as substrates. So, extra 2 phosphates provide this energy of polymerization. Now let us see second scenario. Here, long segment of DNA has to be replicated. Separation of long segment of two strands of DNA require high energy for enzymes to start polymerization from two ends. Instead, only small part of DNA is separated. This is called replication fork. When DNA polymerase enzymes work in replication fork, two types of new strands are formed: continuous strand and discontinuous strand. Discontinuous strand is joined by DNA ligase. DNA polymerase cannot initiate process of replication by themselves, and they cannot initiate at random places. There is definite region in E. coli DNA where replication starts. Such region is called origin of replication. Remember stages of cell division. DNA replications occurs at S phase, and DNA replication and cell division cycle should be highly coordinated to get normal daughter cells. If cell division fails and replication occurred, it results in more DNA in single cell. The condition is called polyploidy. Now let us discuss about transcription. Here, genetic information is copied from DNA to produce RNA. In place of thymine, here uracil is added. In replication, Whole DNA is copied and replicated. But in transcription, only part of DNA and the two single strand is copied. So, region to be transcribed have a defining sequences on either side. Why both strands are not copied in transcription? Because enzymes of protein synthesis read information from single RNA strand. If two strands are copied, two RNA strands are formed. Both are complementary and form double-stranded RNA. Protein synthesizing enzymes cannot read this information from double-stranded RNA. If they don't form double-stranded RNA also, two different proteins are formed from two RNA strands formed. Means single segment of DNA result in synthesis of two different proteins. Now, let us imagine a book with 10 chapters in it. Replication is like getting whole book photocopied. Transcription is like getting only one chapter in that book photocopied. Let us think you want to photocopy fifth chapter. How will you know where it starts and where it ends? Obviously, there will be chapter title to find it and summary at the end of the chapter. Same way, when only one part of DNA is transcribed, it has other regions which guide enzymes. Together, they form transcription unit. So, transcription unit contains promoter, the structural gene, and a terminator. Now we know that only one strand is copied during transcription. Some convention is followed to avoid confusion. Strand with polarity 3' to 5' is template strand. Strand with polarity 5' to 3' is coding strand. DNA dependent RNA polymerase read template strand and produce RNA which resembles coding strand. Promoter, structural gene, and terminator are described with respect to coding strand. So promoter is located towards 5' end of coding strand and terminator is located towards 3' end. RNA polymerase binds to promoter region and reads information in template strand. So promoter is upstream to the structural gene and terminator is downstream to structural gene. Apart from promoter and terminator, other regulatory sequences may exist upstream or downstream. Now let us understand easily the words polycistronic, monocistronic, exons, introns. Cistron is part of gene which actually translated to protein. Let us understand these with respect to promoter and terminator region. If one cistron is present in between promoter and terminator, it is monocistronic. It is seen in eukaryotes. If multiple cistrons are present in between promoter and terminator, it is polycistronic, which is seen in prokaryotes. Again, monocistron of eukaryotes have exons and introns in it. Exons are parts which expressed and introns are parts which are not expressed. Now we will see types of RNA and process of transcription. First, we will see transcription in prokaryotes or bacteria. There are three major types of RNA. mRNA, tRNA, rRNA. mRNA acts as template. tRNA reads and brings amino acids. Again, tRNA reads mRNA and bring amino acids. rRNA provide place for mRNA and tRNA to attach. So it plays structural role and also forms peptide bonds between amino acids. So it also plays catalytic role. Let us see the process. RNA polymerase comes and opens the two strands of DNA. RNA polymerase, along with the initiation factor, binds to the promoter region. RNA polymerase synthase RNA strand by elongation. When it reaches terminator region, it binds with the termination factor and finishes transcription. Prokaryotes have only one type of RNA polymerases for all RNA synthesized. One more thing to note is RNA formed in prokaryotes doesn't require any further processing. So, translation also starts before completion of transcription. And both process takes place in same compartment of cell. Let us see transcription in eukaryotes. There are three types of RNA polymerases in eukaryotes. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Let us see about RNA polymerase 2. RNA polymerase 2 is responsible for transcription of mRNA or formation of mRNA. First, here heterogeneous nuclear RNA is formed, that is hnRNA, which require further processing because it contains both exons and introns. So, it is subjected to 1. Splicing, where introns are removed and exons are joined. 2. Capping, where methyl guanose and triphosphate is added at 5' end. 3. Tailing, around 200 to 300 adenylate residues are added at 3' end. Now this is fully processed RNA. Now it is called mRNA. So here hnRNA is converted into mRNA. RNA polymerase 1, it helps in the transcription of rRNA or ribosomal RNA and also 28S, 18S, 5.8S subunits. RNA polymerase 3 helps in the transcription of tRNA, 5-SrRNA, and snRNA. Now let us discuss about the next topic, genetic code. We know that ultimately information present in nucleic acid is passed to form amino acid sequence, that is protein. It was George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because a permutation combination of 4 power 3 would generate 64 codons, generating many more codons than required. He had given proposition. Now it has to be proved. Hargobind Khurana developed chemical method to synthesize RNA with defined combinations of homopolymers and copolymers of nucleotides. It was instrumental. Marshall Nirenberg's cell-free system for protein synthesis finally helped to code to be deciphered. Severa-Ochoa enzyme, which is also called as polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in template independent manner. That is, without template, synthesis of RNA could be possible with this enzyme. So finally, genetic code is triplet of nucleotides is proved. That is, group of 3 nucleotides gives information about amino acid. The salient features of genetic code are as follows. The codon is triplet. That is, 61 codons code for amino acids and 3 codons are stop codons. They do not code for any amino acids. One amino acid can be coded by more than one codon. This is called degenerate. The codon is read in mRNA in a continuous fashion. There are no punctuations. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. AUG codon has dual functions. It codes for methionine and it also acts as initiator codon. UAA, UAG, and UGA are stop or terminator codons. Now let us discuss about mutations. We will see about point mutation. If one base pair is removed or added, it results in frame shift mutation. If two base pairs are added or deleted, it also results in frame shift mutation. If three base pairs or multiples of three base pairs are added or removed, reading frame does not change from the point of mutation. We know that information in mRNA is in the form of nucleotides. Proteins are synthesized from that information, which are amino acids. So, Francis Crick proposed that there should be an adapter molecule or common molecule which could read information on mRNA and also help in synthesizing polypeptide chain. Yes, he was correct. There is a adapter molecule. It is none other than tRNA. tRNA has an anticodon loop that has bases complementary to the code, and also it has an amino acid acceptor end to which it binds amino acids. tRNAs are specific for each amino acid. For initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNAs available for stop codons. tRNA is said to be cloverleaf or inverted L shape. Now, let us study about translation. What happens in translation? Here, polymerization of amino acids occur to form a polypeptide chain. mRNA, rRNA, tRNA are involved in this process. Now imagine there is a sequence in mRNA. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and stop codon, and it codes for a polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated regions. The untranslated regions are present at 5' end, that is before start codon, and at 3' end, that is after stop codon. They are required for efficient translation process. The ribosome is cellular factory, which consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of peptide bond. The ribosome also acts as a catalyst. Example, 23S rRNA in bacteria is the enzyme. This enzyme is called ribosine, and it is used in the formation of peptide bond. Amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. For initiation, the ribosome binds to the mRNA at the start codon, that is AUG. It is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of amino acid linked to tRNA sequentially bind to the appropriate codon in the mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequence on the other side. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. The amino acids are joined by a bond which is known as peptide bond. Now let us discuss about regulation of gene expression. Gene expression is essentially the process by which a cell decides which proteins to make and when. Think of it like a factory. Just because the factory can make 100 different products, it doesn't mean it should make them all at once. It only produces what is needed based on current demand. In complex cells like eukaryotes, there are four main checkpoints where the cell can stop or start the process. First is at transcriptional level. It decides whether to copy DNA into primary RNA. Second is at processing level. It decides whether to edit that primary RNA, that is, splicing, to make it readable. Third is at transport level. It can control the movement of the finished RNA, that is mRNA, out of the nucleus to the cytoplasm. Fourth is at translational level, where it can control building the protein from the mRNA. Why regulation happens? Cells don't waste energy. They express genes based on their environment, metabolism, or growth needs. Let us take a environmental example. E. coli bacteria only produce the enzyme beta galactosidase if lactose is present. If there is no lactose to break down, the bacteria turn off the gene to save energy. Let us take another example of developmental example. The way an embryo grows into an adult is just a highly coordinated schedule of turning different enzymes on and off at the right time. Now let us see how gene regulation works in bacteria or in prokaryotes. In simpler organisms like bacteria, the main control point is at the very beginning, that is at transcriptional initiation. Let us see or let us think RNA polymerase is the machine that reads the DNA. Promoter is like the start line on the DNA where the machine sits down or RNA polymerase sits down. Regulatory proteins act like traffic controllers. There are activators. They are like green light. They help the machine to start. And there are repressors, which are like red light. They block the machine from starting. That leads us to the operon system. Bacteria organize related genes into a unit called an operon. Operator region is a specific switch located next to the promoter. Here, each system is unique. Example, a lac repressor only fits the lac operator, much like a specific key fits a specific lock. If the repressor is sitting on the operator, then the gene is off. Let us see about the lac operon. Lac operon is a fascinating biological switch found in bacteria E. coli. It allows the cell to produce the enzymes needed to digest lactose when lactose is actually present. Think of it as a smart home system that only turns on the heater when it detects the temperature has dropped. The operon is a cluster of genes that work together. You can break them down into two groups: structural genes and regulatory genes. There are three main structural genes that help in breaking down lactose. One is LACZ. It makes beta galactosidase, which digests lactose into simple sugars, glucose and galactose. Second is LAC-Y. It makes permease, which acts like a doorway to let more lactose into the bacterial cell. Third is LAC-A. It makes trans-acetylase, that is, it assists in the process. And there are three regulatory genes. One is I gene. This is the inhibitor. It works 24 into 7 to create a repressor protein. Second is promoter. It is a landing strip where RNA polymerase starts. Third is operator. It is a on and off switch located right next to the genes. Let us see how lacoperon works. We will discuss it in two scenarios. The first scenario is when there is no lactose. When there is no lactose around, the I gene produces the repressor. The repressor binds tightly to the operator like a physical roadblock. RNA polymerase tries to move forward to transcribe the genes, but it gets blocked. The result is no enzymes are made. Now, the second scenario is when lactose is present. When you feed the bacteria lactose, the system switched on. Lactose, which is the inducer, enters the cell. Lactose molecules bind to the repressor protein. This changes the shape of the repressor, making it let go of the operator. Now the roadblock is gone. RNA polymerase can now slide down the DNA and transcribe the genes. The result is the cell produces the enzymes to eat the lactose. Key concepts to remember from lac operon are: first, it is a negative regulation. This system is called negative because the default state is blocked by a repressor. You have to remove the block to make it go. Second, it is a constitutive expression. The I gene is always on. The cell always keeps a few repressors and a tiny amount of permease just in case lactose shows up. Third is about the inducer. Lactose is its own inducer. It is the very substrate the enzymes are meant to digest, making the system incredibly efficient. Now let us discuss about Human Genome Project. Every living thing has a unique genetic code found in its DNA. If two people are different, it's because their DNA sequences are different. This simple fact sparked a massive quest. If we could map the entire human genome, we could finally understand the instruction manual for a human being. In 1990, thanks to new tools in genetic engineering, the Human Genome Project was officially launched. It is a mega project. To understand the scale of this project, you have to look at the numbers. If you see the size, the human genome contains about 3 billion base pairs. If you see the cost, at an initial estimate of $3 per base pair, the total cost was roughly $9 billion. If you see the data, if you print this sequence in books with 1000 letters per page and 1000 pages per book, you would need 3,300 books just to store the DNA of a single human cell. Because of this mountain of data, scientists had to use high speed computers for storage and analysis. This gave birth to a new field of biology called bioinformatics. The project was not just about reading code, it had specific goals or targets. First, sequence all 3 billion chemical base pairs. Second, identify all 20,000 to 25,000 human genes. Third, store this data and create better tools for analysis. Fourth, address the LC, that is, ethical, legal, and social issues that come with knowing our genetic secrets. If you see cooperation from different countries, the Human Genome Project was a 13-year international effort. It was led by the US Department of Energy and National Institutes of Health, with major help from the UK's Wellcome Trust, as well as teams from Japan, France, Germany, and China. The project reached its primary completion in 2003. Scientists used two main approaches to tackle the DNA. Approach 1 consists of expressed sequence tags. Here, they focused only on the genes that are expressed as RNA. Approach 2, which is a sequence annotation. It is a blind approach where they sequenced everything, that is, whole genome, and figured out which parts were important later. Since DNA is too long to read in one go, they broke it into small random fragments. These fragments were put into vectors like bacterial artificial chromosomes and yeast artificial chromosomes to be copied and amplified. It is called cloning. They used automated machines based on a method by Frederick Sanger for sequencing. Computers then looked for overlapping parts of these fragments to stitch the whole sequence back together, much like a giant jigsaw puzzle. While most of the project finished in 2003, chromosome 1, the largest one, was not fully sequenced until May 2006. To finish the map, scientists also used markers like microsatellites. Microsatellites are repeating DNA patterns. They used these markers to help pinpoint where specific genes are located. By understanding DNA, we can now find better ways to diagnose and treat thousands of disorders. We have also sequenced other organisms like bacteria, yeast, fruit flies, and rice to help us solve challenges in healthcare, agriculture, and the environment. Key findings of the Human Genome Project are: our DNA is composed of roughly 3.16 billion base pairs. We have about 30,000 genes. This was a shock to scientists who originally expected to find over 1 lakh. An average gene is about 3,000 bases long. However, some are massive. The dystrophin gene, for example, spans 2.4 million bases. Humans are incredibly similar. On a genetic level, 99.9% of our DNA is identical across all people. Only a tiny fraction, less than 2% of our genome actually contains instructions for making proteins. For over half of our genes, we still don't know exactly what they do. A huge portion of our genome consists of repeated sequences. These are stretches of DNA that repeat hundreds or thousands of times. While they don't code for proteins, they help us understand how chromosomes stay stable and how we evolved. If you look at individual chromosomes, chromosome 1 is the busiest with nearly 3000 genes, while the Y chromosome is the smallest contributor with only 231 genes. Scientists found 1.4 million spots. where humans differ by just a single DNA letter. These are called SNPs, pronounced as SNPs. The full form is single nucleotide polymorphisms. These SNPs act like markers to help us find the cause of disease and track human history. Now we will see applications of Human Genome Project. The completion of Human Genome Project marks a turning point in how we do science. Let us see the old way versus new way. In the past, researchers were like explorers, looking at one tree at a time. Now, with the full sequence, we can see the entire forest. Instead of studying one gene in isolation, we can look at how tens of thousands of genes and proteins work together as a giant interconnected network. The impact of Human Genome Project is it leads to medical revolution. We can now study all the active genes in a specific organ or even within a tumor to understand exactly how diseases work. Turning this raw data into meaningful knowledge is the great challenge of the coming decades. It will require tens of thousands of scientists from across the globe working together to decode the chemistry of life. Let us see what is DNA fingerprinting. Imagine you have a library of 3 billion letters. Even though 99.9% of the letters in every human's library are identical, that remaining 0.1% still leaves 3 million differences. Sequencing all 3 billion letters every time we want to compare two people would be impossible. Instead, we use DNA fingerprinting, a shortcut that looks only at specific unique landmarks in our genetic code. Let us see about the satellite DNA. Most of our DNA codes for proteins, but a large portion is junk or non coding. This non coding DNA contains repetitive DNA, where short sequences are repeated many times. When we spin DNA in a centrifuge, the bulk DNA stays together, but these repeats show up as separate smaller peaks called satellite DNA. Because these repeats don't usually affect our health, mutations here just pile up over generations without causing harm. This creates polymorphism, that is, huge variations between people. Let us see how DNA fingerprinting works. Now, the secret weapon of DNA fingerprinting is VNTR, that is, variable number of tandem repeats. What is it? It is a mini satellite where a small sequence is repeated over and over. One person might have 10 repeats at a specific spot on a chromosome, while another person has 50 at same spot. Since you inherit these repeats from your parents, they are perfect for paternity tests and forensic identification. Every person has a unique pattern except for identical twins. How DNA fingerprinting is done? Initially developed by Alec Jeffries, the process follows these clear steps. Step 1, isolation. Here, the DNA is extracted from a sample, that is, blood, hair, or even a single cell. Step 2, digestion. Here, we use molecular scissors, like restriction enzymes, to cut the DNA into fragments. Step 3, electrophoresis. Here, the fragments are run through a gel. Smaller fragments move faster and further, separating them by size. Step 4, blotting. Separated DNA is transferred from the fragile gel onto a sturdy nylon sheet. Step 5, hybridization. Here, probes are added, that is, a radio labeled known VNTR that seeks out and sticks to its matching sequence on the sheet. Step 6 is detection. It is detected by using X-ray film or autoradiography to see where the probes are stuck. This reveals a unique barcode of bands. DNA fingerprinting is not about reading your entire genetic book. It's about checking the unique status in your code. By using VNTRs as markers, we create a genetic barcode that can solve crimes, settle paternity disputes, and track human evolution, all from a single cell's worth of data. In the early days, you needed a large, fresh sample of DNA to get a result. But today, thanks to PCR, forensic scientists can now create a full DNA fingerprint from an old bone, a tiny drop of dried saliva, or a smudge of skin cells left at a crime scene. PCR acts like a molecular photocopier. It takes a tiny microscopic amount of DNA, even from a single cell or a single hair follicle, and amplifies it, that is, makes millions of copies.